Today about the development of a novel I-like reporter gene cell line for the quantification of neutralizing antibodies against recombinant AAV vectors. But first a word about adeno-associated virus. AAV is a 4.8 kilobase single-stranded DNA parvovirus, and the AAV genome encodes 3 genes: rep for replication, cap for capsid, and ap for assembly, and these give rise to 9 gene products, including the assembly activating protein, which is encoded by the ap gene in a different reading frame within the cap gene. And the AAP protein is essential for capsid assembly, at least for some serotypes, including AAV3. AAV replication is defective and requires either live adenovirus or some other means to supply the immediate early enhancer proteins required to replicate the virus or indeed to express a transgene. The host immune response during administration of recombinant AAV vectors can limit long-term transgene expression, and this has been shown in a number of clinical trials. Most of the world's population has been exposed to wild-type AAV, often in early childhood, and it's been shown in the normal European population that some 70% of individuals present antibodies against both AAV1 and 2, and 50% or so against 8 and 9, but there's a high degree of cross-neutralization between the different antibodies, so an antibody against AAV2 can neutralize AAV8 and 9, etc. So this pre-existing immunity and memory effect can result in a loss of transgene expression, often within 6 to 8 weeks following initiation of treatment with a recombinant AAV vector. And it's been shown with factor 9 that there's a temporal correlation between both a humoral and a cellular response against the capsid proteins. But there are also antibodies produced, but less frequently, against CpG sequences within the virus genome. So priming and expansion of a pre-existing antigen-specific immune response can activate both a humoral response and also a CTL, cytotoxic lymphocyte response, which can lead to clearance of transduced target cells. So it's obviously of importance to develop suitable assays for the quantification of neutralizing antibodies and also a cellular response to AAV immunogenicity. This shows you the basic setup of a recombinant AAV vector. You have the gene of interest, and this is the expression of which is regulated by a promoter. This can be a constitutive promoter or a tissue-specific promoter, and obviously, depending upon the target for expression of the transgene, this could be, for example, a retina-specific promoter if the transgene is to be expressed in the eye, or a fibroblast-specific tissue promoter, etc. This is followed by a terminator, a suitable poly A, all this within the inverted terminal repeats, and again within about 4.8 kilobases. This shows a typical packaging cell line for the production of a recombinant AAV vector, and all the molecular components shown here can be provided either by transitive transfection or preferably integrating the components into the genome of the cell line. The host cell line is chosen as a function of both its safety profile and also its ability to withstand the toxicity of the rep protein, which can lead to cytotoxicity. I'm sure, as you're all aware, all preparations of recombinant AAV vectors, including those administered to patients, contain, in addition to whole virus expressing a transgene, often a large excess of empty capsids, individual capsid proteins, naked non-encapsulated viral DNA, and then various cellular components from the packaging cell. And all these components are potentially immunogenic. If you look at this gel on the left-hand side, you can see after cesium chloride density ultracentrifugation, this is obviously not a methodology that can be used on a large scale, or indeed for clinical material, as cesium chloride is a neurotoxin. But even after this very sophisticated purification process, you can readily see some of the capsid proteins, VP1, VP2, VP3, and on the extreme right hand of the slide, even after standard ion exchange chromatography, there are a myriad of proteins remaining in this AAV preparation. So in order to develop the dual reporter gene system that we've developed, you need two essential components in the packaging cell. We've added a tag. This is a GAL4. GAL4 is a yeast protein which does not exist in mammalian cells, conjugated with a VP16, it's a transactivator from HSV1, and these are again all the elements that you need for the packaging cell line. So in order to reduce the excess of empty capsids, we've expressed the cap gene under the control of an inducible promoter. This is the Van-O promoter here. And by regulating the expression of the cap protein relative to the transgene, you can reduce very markedly the number of empty capsids. So the principal requirements for this dual assay are the packaging cells must contain the immediate early enhancer proteins required to express the transgene encoded by an AAV vector and express a specific capsid transgene combination, which we've shown you. The reporter cells must also respond in a specific manner to a given capsid transgene combination. This shows you a reporter gene construct with a 5 times tandem repeat of the GAL4 upstream activation sequence to which the GAL4 VP16 tag will bind in a highly specific manner. The fact that GAL4 is a yeast protein, this renders the assay extremely specific. This shows a variation on the reporter gene cell line. Here we've added a normalization gene. We've shown previously that that normalization of drug-induced firefly luciferase activity relative to the constitutive expression of a second luciferase gene, this is Renilia, under the control of a constitutive promoter, renders the assay independent of cell number and provides a very useful way for compensating from serum matrix effects. If you mix the packaging cell line with a reporter cell line, the virus will become excreted from the cell. There's no need to purify the virus or in any manner. You just mix the packaging cell with the reporter cells. The virus will then attach to the surface of the reporter cells initially via sugars. For example, AAV2 is primarily heparin sulfate. It then combines with the first cellular receptor. This is the receptor described by Pili in 2017 for the first time. And depending upon the serotype, the virus will attach to different domains of this receptor. The virus then becomes attached to the endosome and attaches to a second receptor. The virus becomes encapsulated, and the virus genome via the GAL4 VP1 tag will bind to the upstream activation sequence and activate firefly luciferase. In the absence of an AAV antibody, you will get activation of both the AAV-activated firefly and the constitutive expression of the Renilia luciferase. In the presence of neutralizing antibodies, however, the response of firefly will be reduced. However, that of Renilia will remain the same unless, of course, there is some non-specific toxicity, and then results can be normalized relative to this. This shows the relationship between the reporter gene signal and the number of reporter cells. We usually use a report of 1 to 15. And again, this shows you the packaging cell reporter cell ratio. And again, as I said, 1 to 15. This has a very good dynamic range. As shown in this assay, it's something like 500-fold. This slide shows you a titration of an anti-AAV neutralizing antibody, and you can see there's a very good dynamic range, and it's a highly sensitive assay. Here we've either pre-incubated the serum sample to be analyzed with the packaging cells for 1 hour prior to addition to the reporter cells, or we've added the serum sample directly to the packaging cells and reporter cell mix. And as you can see, there's no difference, so obviously the easiest way to do things is simply add your sample to a mixture of the packaging cells and reporter cells and incubate overnight. This shows in the blue the SPA dual reporter gene assay relative to the reference test. This is a transduction inhibition assay based on an AAV2 vector expressing far-field luciferase. And this is a 3-day assay which initially involves incubating the virus containing the luciferase gene with the sample to be detected and then incubating for 3 days with an HEK cell-responsive cell line. So as you can see, our assay is more sensitive and has a better dynamic range. So to conclude, we've developed a highly specific capsid transgene combination. This is some 10-fold more sensitive than the transduction inhibition reference assay. It's rapid overnight incubation as opposed to 3 days. And it can be used to screen antibodies to recombinant AAV vectors, both to AAV2, 5, 6, 8, and 9, and also hybrid and chimeric capsids. There's no need to purify or isolate the virus, and this is available in a frozen thaw and use format, so all you do is thaw a vial, add your sample at a suitable dilution and incubate overnight, and then read the result. That's it. Thank you very much for your attention.